Hello everyone, can you hear okay? Good, lots of thumbs up. So yeah, I'm Adam Roberts, I'm a postdoctoral researcher at UKHSA, and just a little bit about my PhD today, which seems many years ago now, but obviously still within the ECR bracket, I promise you. But that's over at Brighton and Sussex Medical School where I did that. So yeah, let's just get into it. I think it's just a good example, I think, of where there's target-based approaches to AMR. And I think that's sort of important to bear in mind when we're looking at, you always want to screen lots of drugs, but it's always good to know what targets we're hitting and how effective they will be from that side. So as we know, with this Victorian disease, which is still very much here, I'd say it's, there's tens of millions of people infected with TB every year. It's a massive global burden. And obviously, that sadly, people succumb to that disease as well in the millions as well. The problem with TB as well, it's a really hard disease to treat. So 6 months of 4 antibiotics. These are toxic drugs, these are horrible drugs, generally people don't like taking them, so that worsens the problem, and then we have, on top of that, huge amounts of multi-drug resistance as well, nearly 500,000 there every year, which is awful, really, but I think, the thing is that's characterised by being resistant to rifampicin, and also isoniazid generally as well. Now this is a drug that's going to be the focus of the talk today. It's quite a small molecule, but I think it's got a very powerful early bactericidal activity, which I think we should try and preserve, but there is drawbacks to this as well. So, what was my PhD on? Obviously, we just heard about a lot of sort of genomic applications to AMR. I looked at more the phenotypic mechanisms as well. So, TB itself has a very thick and waxy cell wall. I mean, obviously, cell walls are quite thick anyway, but for TB, it's just very strange. It literally is like a candle of a bacteria. It's very, it's very impervious to a lot of drugs already. But there's more active mechanisms, as you can see in this really well-drawn graph here. Who needs biorender? I think, it does pump out a lot of things as well, but obviously, the main thing being antibiotics in this context. So my PhD was to go and characterise loads of efflux pumps, off you go, and I was like, oh, thanks. But then I came look at this one particularly in efpA, the imaginative name of efflux pump A, but this is efflux pump A is efpA is very much essential to the growth of TB, so I can't grow without it. It's very necessary. When I started my PhD, there wasn't a lot of information apart from it being essential, but since my PhD, obviously things have come out and said it's probably involved in the transport of cardiolipin, which is actually quite weird for efflux pumps to actually transport as part of this family. But the main thing we're interested in is the context of drug resistance and tolerance. So efpA expression is increased when TB is exposed to isoniazid. So there's a sort of question there. You can see whether is efpA required for the resistance and or tolerance to isoniazid? So is it involved in that? But obviously, I've gone about how it's essential. How do we study an essential efflux pump? Obviously, everyone's wanting to know the answer to that question, and that's generally through CRISPR interference. This obviously wasn't my whole PhD, I was doing very short, but this is sort of the real, the bare bones part of it. But CRISPR interference is a brilliant system because it allows you to sort of dial down the expression of your gene of interest. So we can't knock it out, so let's just dial it down so the TB is obviously not very happy. But at least it's there, and it can induce this system with the drug anticyclin. I'll just call it ATC because it's a mouthful. But this is a great system because you can essentially just pop the CRISPR Cas system here on your gene of interest, and that will then block transcription. And how much drug you add is how you can basically tune how much knockdown you want of that gene. And so that's what the great thing about that is. After that, we're looking at the sort of confirming that the efpA knockdown by RT-qPCR and just seeing what effects it has on viability and also some morphology through SCM as well. And once we had those down, then obviously looking at exposing that knockdown strain, so once the TB's obviously a bit affected by the knockdown of that essential efflux pump, we can then affect it with isoniazid and then see if there is an interaction between the two. So going with the data, our first graph, just showing the viability, has been that all these, basically there's 2 strains, there's a vector control and a knockdown strain. The vector control is just the CRISPR system is on, but it's not targeting anything, it's just random junk DNA essentially. But then the efpA knockdown is targeted to efpA, so it will knock down efpA. So when it's induced, that's what this purple dotted line here is showing, there is a massive reduction in viability, which does recover because the system doesn't last forever once it's induced with that drug. And then obviously I had to prove that indeed it was knocked down and found that through the RT-qPCR, there was indeed a 43-fold reduction in the expression of efpA by day 1. Obviously that did recover, but by that point it seems to have had the effects which we desired, that it has knocked down that efflux pump and it's having an effect on the cell. And that sort of corresponds here, that it starts to recover after that. So they do sort of line up in this data set here. So there are some weird sort of morphological changes. So this is a normal graph of TB. They are a bit dehydrated, but these are essentially the same length here. And it'll be about 2 to 3 microns. They're all quite uniform. That's one thing to be sort of aware of. But then once you knock down the efpA, they become very lengthened. So there's some of them up to 9 microns in length. And there's also, to know, there's quite a lot of small cells as well. And that's because I've noticed when measuring these cells, there's quite a big variability in terms of the cell length now. So maybe an efpA might be involved in cell division as well, or at least the sort of the normal function of that system. So I did talk about isoniazid, I haven't forgotten, but this essentially is a really good drug because there's a really powerful early bactericidal effect, both in vitro and in vivo. There's 3 studies here, just an example. We have something from our lab here, which is a chemostat model. Isoniazid is given at 16 times MIC, but there's always a recovery. And there's in vitro here as well. We have different concentrations of isoniazid, and then here in a sort of mouse model as well, we can see that there is isoniazid here in this line here. But you can spot every time there's a regrowth. Isoniazid is great initially, knocks down the viability, and there's always a regrowth, and that's a massive problem for this great drug. So, is efpA involved in that recovery of viability at TB following isoniazid exposure? That was the main question that I was seeking here. So, I decided to, do a really long experiment, 45 days, because TB is insane and takes 24 hours to replicate, to split. So, what you're seeing here is the same graph before, but over a longer time period, where we can just check out the system, and that is indeed a knockdown of viability. Then here we have, in the orange, the 2 strains are exposed to isoniazid. We have a really good bactericidal effect, like 3 logs reduction or so, maybe even 4. And then, it comes back after, and that's a real problem, because obviously those patients who would just have that mono, they don't obviously give them mono, but at this point they would obviously be sick again. What happens if we knock down efpA and give it isoniazid? It goes completely the other direction. So basically near clearance of the bacterial viability here. So it completely just plummets, cannot function whatsoever. And even after 45 days, there's basically no CFU which is detectable. They're very much sitting on the limit of detection if they are there. So obviously it's having a massive effect. So we can conclude that repression of efpA does prevent the regrowth of TB exposed to isoniazid as well. So that was the main focus of the PhD. But yeah, I just wanted to say, we'll summarise what we just saw. But I think just to conclude, I would say that efpA could be a very good viable target for combination with isoniazid therapy. Independent studies, such as that by Johnson et al. In Nature, a huge chemical genetic screen had landed on efpA as one of the best targets to target in TB to affect its viability. So not only is it going to be helpful for isoniazid resistance, to prevent that happening, but also it's a good target in its own right just because of its sensuality. And there are already inhibitors which exist, like BRD 8000.3, which we could use hopefully at one point in conjunction with isoniazid to have a good effect. So thank you very much for listening. I'll have to take any questions.